G-protein coupled metabotropic membrane receptors. There are three different classes of membrane receptors: G-protein coupled receptors, ion channel receptors, and enzyme-linked receptors. In this presentation, we will be focusing on the G-protein coupled metabotropic membrane receptors. Membrane receptors are specialized proteins on the cell membrane that allow for the communication of signals in and out of the cell. These signals are communicated to a cell through what is called a signal transduction pathway or cascade. Also, the G-protein coupled receptor in this video will activate the second messenger of cyclic AMP in order to accelerate metabolic activity of the cell. Since G-protein coupled receptors indirectly affect membrane potential and they act through these second messengers, they are known as metabotropic receptors.
Step 1. To begin signal transduction in the G-protein coupled receptor, it begins with a ligand binding to the receptor protein. A ligand is a substance that binds to the receptor to communicate a signal. There will be two messengers in this receptor, but this is the first. The ligand in the G-protein receptor can be a neurotransmitter or a hormone. Some examples of neurotransmitters or hormones that bind to this type of G-protein receptor include epinephrine and calcitonin.
Step 2. The binding of the ligand to the receptor causes activation of the G-protein complex, which is attached to the receptor. The G-protein is heterotrimeric, meaning it is composed of three subunits, the alpha, beta, and gamma subunits. These are G-protein because they bind to GDP and GTP. When activated, the G-protein converts GDP or guanosine diphosphate into GTP, guanosine triphosphate.
Step 3. With GTP attached, the alpha subunit then detaches from the other subunits and binds to an enzyme called adenylkylase.
Step 4. Inside the plasma membrane, adenylene cyclase then converts ATP or adenosine triphosphate into CAMP or cyclic adenosine monophosphate, which then increases levels of cyclic AMP inside the cell.
Step 5. Cyclic AMP is a second messenger that typically activates protein kinase A, or pKa. Kinases phosphorylate proteins in the cell, which means they attach a high-energy phosphate group to proteins and changes their structural conformation.
Step 6. These phosphorylation reactions by protein kinase A can mediate both short and long-term cellular responses in the cell depending on the proteins they phosphorylate. For example, protein kinase A can phosphorylate enzymes inside the cytoplasm, which can cause many kinds of responses, one of them being acceleration in metabolic activity.
Step 7. In order to stop the cellular response, cyclic AMP levels are decreased by an enzyme called phosphodiesterase, or PDE, which inactivates cyclic AMP by converting it into AMP, or adenosine monophosphate. This ends the signal transduction cascade in G-protein coupled membrane receptors using the second messenger cyclic AMP.